Good day, ladies and gentlemen. The paper I'll be presenting is actually my undergraduate thesis back at Batangas State University, Alangilan campus, a couple of years ago. But actually, I still find it timely and relevant, especially with the current environmental situation we are in. Allow me to introduce myself. I am Cheryl D'Agustin. Currently, I am a registered chemical engineer and taking my master's degree on environmental engineering at Papua University as a TOSTERTT scholar. The other proponents of of this research are Engineer Arvin D. Maunahan, Engineer Christian Joy Lapis, and Engineer Ronaldin Maulion as our advisor. So this paper is entitled Utilization of Biosynthesized Magnetic Iron Oxide Nanoparticles with Brown Seaweed's Sargassum Polycystum for the Removal of Methylene Blue Dye. One of the most harmful sectors that contribute to the ongoing environmental contamination is the textile dyeing business. Since most synthetic dyes have complex aromatic molecular structures that are inert and non-biodegradable when discharged into the environment, treatment of effluents such as dyes is a really significant challenge. Many of the industrial dyes are toxic, carcinogenic, mutagenic, and teratogenic. It is calculated that over 100,000 commercially available dyes with more than 700,000 tons of dye stuff are produced every year. Methylene blue is actually a basic cationic dye and is used in human and veterinary medicine for several therapeutic and diagnostic procedures, including as a stain in bacteriology, as a redox colorimetric agent, as a targeting agent for melanoma, as an antidote, and as an antiseptic and disinfectant. In this study, methylene blue dye was subjected for removal in order to produce a clear water. In removing dyes from wastewater, adsorption seemed to be the most efficient method since it is both economical and really effective when it comes to the removal of dyes from industrial wastewater. In general, adsorbent used are smaller in sizes since smaller size adsorbent have increased surface area leading to highest adsorption capacity. But it would be really hard to sort them from water or from the clear water when it reaches saturated adsorption. And in that case, magnetic separation has been one of the most um, promising techniques for wastewater treatment since no contaminants such as flocculants are produced and a large quantity of effluent can be handled within a diminutive period. Nanotechnology has really gained a lot of attention nowadays because of its wide array of functions or uses, particularly this green nanotechnology. This comprises a diversity of processes that diminish or eliminate toxic substances to restore the environment. In particular, green biosynthesis is the term used for the synthesis of metal nanoparticles using inactivated plant tissues, plant extracts, exudates, and other parts of living plants, which is actually a modern alternative for its production. This offers a green approach to the treatment of organic pollutants, which is actually environmentally friendly and uses safe and non-toxic reagents. Magnetic nanoparticles are recognized as efficient adsorbents with large specific area and small diffusion resistance. Also, these magnetic iron oxide nanoparticles, or what we call magnetites, are suitable for removal of dyes because it can be simply removed from water through the process of magnetic separation. Brown seaweed is also an important addition in this study. It has phytochemicals, well, technically it's called sulfated polysaccharides known as fucoidants, which include hydroxyl, carboxyl, and aminofunctional groups, which can serve both as an effective metal reducing agent and stabilizing agent in order to form iron oxide nanoparticles and um, as an effective covering of the metal oxide nanoparticles. Specifically, this study aims to answer the following questions. Number one, how does the varying ratio of brown seaweed extract to ferric chloride solution affect the amount of nanoparticles produced? Number two would be what are the physical properties of the formed iron oxide nanoparticles in terms of its shape, um, particle size and particle size distribution, and pore size distribution. Number three, what is the best condition in terms of the percentage removal of methylene blue dye with varying parameters such as dye initial concentration and contact time? Number four, what isotherm model best suit the equilibrium data? And lastly, what kinetics of adsorption, or well, particularly order of reaction, best suit the kinetics data? As for the methodology, so well, first is the synthesis of iron oxide nanoparticles, which actually is a crucial part of this study. So we collected the brown seaweeds from Barangay Uno, Calatagan, Batangas, where actually they do have brown seaweed farming there. And then the brown seaweed were subjected for freeze drying. It was freeze dried at negative 20 degrees Celsius for a day, um, where we brought the samples to De La Salle, Manila, and then transported back to Batangas State University for immediate experimentation. So the freeze dried samples were crushed into fine powder in order to do solvent extraction. So the ground samples were heated at 100 degrees Celsius with distilled water, and then the synthesis proper of the iron oxide nanoparticles was done using the or when adding the zero point 0.1 molar ferric chloride solution to the brown seaweed extract in a 5 is I mean in a 1 is to 5 volume ratio, so that the iron oxide nanoparticles could be produced. Now, virtually the formation of the iron oxide nanoparticles can be seen through the change in color from a rusty color to a dark brown to dark color, and also the decline in the pH of the solution signifies the formation of the iron oxide nanoparticles. Next is the preparation of the standard textile dye solution, and then the characterization of the synthesized iron oxide nanoparticles, where we actually used SEM, or scanning electron microscope, in order to determine the shape, the particle size, and the pore size of the um, synthesized iron oxide nanoparticles. Next is the optimization of the varying parameters. We actually had two varying parameters here: the initial dye concentration, which are 20 ppm, 30 ppm, and 40 ppm. And for the contact time, that would be 5 minutes, 10 minutes, and 15 minutes. The controlled here are the pH, which is at 11, and the amount of adsorbent used, which is at zero point 0.5 grams. And also, the experiment was repeated thrice to obtain accurate results. And lastly, for our methodology is to determine the suitable model for adsorption isotherm and the kinetics of adsorption. For the isotherm, we used two models, which are the Langmuir and Friedrich models, and tested if our data would suit either of the two. And for the kinetics of adsorption, particularly the order of reaction, we had three models: the pseudo first order model, pseudo second order model, and we actually use also the simple LOV model. Now, for the results and discussions, number one would be the effect on the varying ratio of a brown seaweed extract to ferric chloride solution. Now, from the table that is posted on your screen, we can clearly see that the maximum amount of iron oxide nanoparticles produced is at 5 is to 1 ratio, which is actually 1.22 grams. And then specifically at 6 is to 1, the amount of iron oxide nanoparticles produced is actually lesser, which means that the production reaches its saturation point at 5 is to 1, and beyond 5 is to 1, there it is expected that there would be lesser production of the iron oxide nanoparticles. Now, this section provides the results on the characterization of the synthesized iron oxide nanoparticles. So, using a SEM or a scanning electron microscope, showing on your screen uh, is an image of the iron oxide nanoparticles produced. So, the shape is actually spherical, which conforms to the related literatures of this study. And as for the particle size, it's at 67 nanometer with particle size distribution at 46 to 55 nanometers. And for the pore size, it's 37 nanometer. And for the pore size distribution, it's at 31 to 45 nanometer. These all conforms to the related literatures and actually proves that our formed iron oxide nanoparticles can be an effective adsorbent for the removal of methylene blue dye. Now, this table represents the summary of the percentage removal of methylene blue dye using varying parameters. So as mentioned, we varied the initial dye concentration and the contact time. So from the table, the best condition that we have is at 20 ppm initial concentration and 15 minutes of contact time, which has 88.78% of average percentage removal. So from this, we can say that the absorption amount did not increase proportionally for higher dye concentrations since the active sites were already filled and already saturated. Thus, it is very clear that the percentage removal of methylene blue dye decreased with an increase of dye concentration. So we can say that the initial dye concentration and the contact time has significant effect on the percentage removal of the methylene blue dye. Now, in order to optimize the use of our modified nanoparticles, it is important for us to establish the adsorption isotherm. So the adsorption capacities of the modified nanoparticles were obtained at pH 11 and varying methylene blue dye concentrations. So the data fitted well to the model Langmuir isotherm model actually with the correlation coefficient value of 0.9905 as compared with the value of the Fruitlick isotherm model. These Langmuir isotherm model is based on the assumption that the adsorption corresponds to a saturated monolayer of solute molecules on the adsorbent surface with no lateral interaction between the adsorbed molecules. And lastly, to better elucidate the reaction mechanisms and understand the absorption diffusion form, we use three kinetic models, namely pseudo first order, pseudo second order, and simple Elovit models, for us to also determine the rate controlling step for the entire reaction during the absorption experiment. Okay. Um, Here is the summary of the values for each, or I mean for all the models that we used. So the correlation coefficient or the R squared for the pseudo first order kinetic model was relatively low, and the calculated QE value was 0.7310 or 0.73, which do not agree with our experimental data, which is 0.1757. Now from here, we can see that the pseudo second order with the R squared value of 9.9769 and the adsorbed um, amount of 0.1934 adheres to the experimental data of 0.1757, and we can conclude that the entire kinetic data uh, corresponds to the pseudo second order um, kinetic model. This indicates the applicability of the second order reaction or model to describe the absorption kinetics of methylene blue dye using our iron oxide nanoparticles. So the best fit of the pseudo second order kinetic model proves that the mechanism of adsorption was chemisorption via electrostatic interactions or attractions. Based on the presented findings, the proponents or we were able to arrive to the following conclusions. First is that there is a significant effect on the ratio of brown seaweed extract to ferric chloride solution when it comes to the produced or the amount of the produced iron oxide nanoparticles. Second is that the nanoparticles produced are in nanoscale and are capable for high dye adsorption through its relatively small particle and pore sizes. Number three, the best initial concentration and best contact time with the maximum percentage removal of methylene blue dye, 88.78% are 20 ppm and 15 minutes respectively. Number four, that the curve fitted well for the Langmuir isotherm model, which has an R squared value of 0.9905, which proved that there is a monolayer adsorption. And lastly, the mechanism of adsorption is chemisorption via electrostatic attraction, which is according to the kinetics data that well fitted to the pseudo second order model. As for our recommendations, first, it is recommended to consider other species of seaweeds, could be brown, green, or red, for the methylene blue dye adsorption. Second, to evaluate the adsorption of other pollutants such as surfactants and heavy metals using the iron oxide nanoparticles. Third, there is a need to determine the surface area of nanoparticle, which is necessary to determine the number of active sites present. Number four is to study the effect of other parameters such as adsorbent dosage and temperature on the percentage removal of methylene blue dye. And number five is to evaluate more on, say, the antimicrobial property of the iron oxide nanoparticles produced. Now, here are our references. And that's it for my presentation of our research paper. And I'm looking forward to your questions. Thank you very much.